wrapper into my program to see how we can use it. Just brief outline of my talk today, just about sugar beets, and then I'm going to talk about like 4 different projects that we are working, and it's primarily focused on Rhizoctonia today, so I'm not going to go into the details for some of the research projects. But if you think about sugar, right? So everybody loves sugar, right? From the time you get up, coffee, creamer, all the way up to, looking for dessert, right? So I was hungry. But as you know, most of the sugar from the temperate region comes from sugar beets. And if you go a little bit south, especially Louisiana and Florida, so most of the sugar comes from sugarcane. So if you think about the numbers, we get about 50% of the sugar in the US comes from sugar beets and 50% from the sugar cane there. When it comes to sugar beets, I think it has changed since 1800s, how and where we grow sugar beets, but this is the most current situation. Like I said, most of the tropical region has the sugar beets. And then this is about the percentage of the contribution in the US. So between Minnesota and North Dakota, we have about 55%, 54% of the sugar beets that are produced. And then we have about 1.2 million acres in the US total. That's the total sugar beet acreage. So it's a pretty significant contribution from this bi-state area here. And the other major ones were actually the Saginaw Valley in the Michigan here, and then Idaho, and then we see some other in the Great Plains areas too, some factories, and there also the sugar beet production. And if you think about Minnesota and North Dakota, we have about 650,000 acres. Primarily, we have 3 cooperatives. The one up north here, that's American Crystal Sugar Company. And then if you go further south, that kind of central Minnesota here, that's Mindak Farmers Co-op. And then one just completely based in Minnesota, it's a Southern Minnesota Beet Sugar Co-op. Between 3, we have 7 factories that are operating. And in terms of contribution to the economy, it's about $4.5 billion. So that's a pretty significant chunk of money for the 2 states just from the sugar beets. It's a vibrant crop, and the growers are pretty active in growing this. So I have to look back my numbers, but I think Iowa, there used to be some sugar beet acres in Iowa, I think a long time ago, so not anymore. And then I don't know how many of you seen sugar beet crop, but that's a mature sugar beet. I mean, basically, the cross section of the soil, you have these nice tops. I mean, very dense foliage there, and it's basically a storage root. So most of the sugars are stored in the root. And then under best conditions, it has about 17 to 80% of sugar. So that's the best crop you can get. And the average tonnage is about 25 to 30 tons per acre. So if you wonder about, that's about 2-pound sugar beet. So if you calculate the sugar, you get 4 Cokes, or you can get 16 donuts. You could just have that much sugar in 1 sugar beet. So just put some perspective in that. That's a lot of sugar in 1 sugar beet. I used to show the video about sugar beet production, but I was not sure about how the Zoom will handle it. So I'm just gonna go through some of this. It's just like any field crop, and it's not considered as a specialty crop, especially in Minnesota or North Dakota, because the acreage is very high. Even actually in terms of USDA classification, just because the acreage for sugar beets is about 1 million acres, it's not considered as a specialty crop for any kind of funding projects or anything. So it's planted typically in mid-April to mid-May, just for this commercial production. Again, this is for sugar. If you're growing sugar beets for seed production, that's a completely different story. Most of the seed is produced in the Willamette Valley in the Washington state, in Oregon, in Willamette Valley in Oregon. That's where it's done. But just the regular one is actually planted from mid-April to mid-May, and the seed is pelleted, so you use just regular vacuum planters for this. And then since 2009, 100% of the sugar beets are Roundup Ready, so we don't use any of those tough herbicides to take care of the weeds. Just about July 4th, we enjoy the Independence Day celebrations, but the significant event for sugar beets will be canopy closure. If it grows, like some people say, if you get the corn around your knee length, I think you'll have a pretty good crop by July 4th. So that's a similar thing for the sugar beets there. And then from July to September, the roots expand pretty significantly. And then once the temperatures tend to go down in late August or September, that's really good for accumulating sugars, because if you have really high temperatures towards the end of the season, the sugars are going to go down. Like I said, 25 to 30 tons. That's about 9 to 10,000 pounds of sugar per acre. That's the typical yield that you get. And when it comes to harvesting sugarbeet, there are two phases. Number one, we just need to defoliate and get the tops out of the way here. So he's running the defoliator, and you see these beets lined up nicely. And then you come back with basically a lifter that just going into a trailer here, or you could actually get a truck on the side, and then directly take it to the piling station. So the main harvest of sugarbeet, it's called campaign. It's based on the calendar date. It's always October 1st. You're in Minnesota, so once you hit like October 15, you never know. Everything will be completely frozen, so you can't get anything out. But last year was an exception. We got a lot of rain, and then it took almost 5 to 6 weeks for the growers to complete the harvest. So what do we do once we harvest the beets? They're actually stored in these big piles. We take advantage of all these frigid winter conditions. And these beets are actually stored almost until next June. So it takes about 8 months to process the entire crop. Some of these have, ventilated structures. The airflow is very critical. I'm only 5.6. You can see these piles 25 to 30 feet, pretty high piles that we see. That's just pretty amazing operation to get to see that. Comes to my program. So I'm actually located in the northwest corner of the state. So that's the Northwest Research and Outreach Center. Primarily, the University of Minnesota established these research and outreach centers to cater to the needs of the clients at their local regions. And then this is actually very rich in agriculture, especially some of the small grains, barley and wheat. Now we have not that many acres compared to the North Dakota, but sugar beets is again pretty significant crop here. And right now, so when talk about some of the Rhizoctonia projects are primarily done by Jason. He's a research fellow in my lab, but he just took a position with the American Crystal Sugar. It's been pretty valuable to my program since 2014. And Pratibha did some of the work that I show you very shortly with the fungicide sensitivity, and she just started her PhD at Cornell this semester. And Jake and Samantha, they're working on Aphanomyces root rot. So Jake especially, he's actually developing a detection, like qPCR based detection, and also assembling the genome for Aphanomyces cochleoides with Dr. Corey Hirsch. Samantha will be working on transcriptomics of host pathogen interactions for Aphanomyces. And then we have some Fusarium. It's called Fusarium yellowes of sugarbeet. It's not really a big concern, but we are seeing flare-ups here and there. And I think that the incidence is actually increasing a little bit. And the final one that's actually most significant impact in the past few years is a Cercospora leaf spot. So Austin, he just started his PhD program working on Cercospora. That's kind of snapshot of things going on. And then I can't just emphasize enough, so many undergraduate students that we get, we're pretty awesome to work with. They contribute a lot to the projects, to get things rolling during the year and also during the summer. So the major goal of my program is actually to try to tackle, just number one is, what is that's causing diseases in sugarbeet, right? The growers are better informed, and then try to manage some of the disease management tactics and combining them to integrated management so that they can actually pick and choose what works best for their situation there. So as a part of this, we also do a disease diagnosis, the service during the summer. This is the samples that we get, about 80 to 100 samples every year. But one thing that stands out from this, over the past 4 years is right off the bat, we get a lot of samples that are infected with Rhizoctonia. And then every 2 to 3 years, we get a spike in Aphanomyces. This actually distribution is very similar to the rainfall pattern that we see, because being an oomycete pathogen, whenever we have this, saturated soils, that gets pretty active, and that's basically, season-long problem. And the other situation that we have, especially with sugar beets, when the growers deliver their beets to the piling station, they also get some of the soil that's left on the elevator, and then they have to bring that back and dump into their fields. But when two trucks are dumping the soil, so it's also possible for you to get some of your the neighbor's soil, and then you bring it back. So there's always this mix of these soil-borne pathogens, and that's causing some of the mixed infections. So you see Rhizoctonia Aphanomyces in some soils too. And then Fusarium, like I said, typically low, but we saw a big spike in 2019. It seems that we had this extremely dry period right after the planting. I think that actually stimulated some of the Fusarium infections in those soils there. And then also, whenever it's dry, it's also a challenge for the growers because some of the herbicides won't break down completely, and then they cause a lot of phytotoxicity. When I say the chemical, that's some of the injury that you get. So they're very aware of the problems there. Like I said, this is the acres for the common crops that we grow in Minnesota right now. The trend that we see is we have more soybeans, the green line, and also more corn, just anywhere from corn right from the beginning, but the soybeans really from 80s. I think even from 2000s, there's like steep increase for soybeans, right? Because the commodity price is higher. But what happened was some of the acreage for oats, wheat, that's been taken by the soybeans. So now we have more and more soybeans and corn in the rotation with sugar beets compared to small grains. So that is having some of the implications that we have seen, like why we are seeing higher Rhizoctonia, especially during the recent years there. And also we have some good bit of dry beans actually up north here in rotation with the sugar beets. Not so much in the southern Minnesota. There's more corn and soybean in the southern part of the state. And as you're aware, soybean is a very good host for Rhizoctonia solanae, but I think most of the soybean seedling damping off is whether it's a pre or post emergence, it's caused by anastomosis group 4. But especially here, we have AG2-2. That's also a very good host for soybeans. So everything I'm showing here is actually infested with AG2-2 here. Again, some of the root rot on the seedlings. And then this is part of the Pretibus project. She was actually evaluating several soybean germplasm for resistance to Rhizoctonia. She did an awesome job that she killed almost 99% of the plants in the furrow when she put the Rhizoctonia that's grown on the barley. And then these are non-inoculated plots. So, but thing is, when we planted this trial, there was about 1 inch rain right the very next day, and it was a little bit later planting, then you could see the impact on the soybeans. But in a commercial field, we don't see this much damage. Again, the trial was repeated in 2019. It's not as severe as 2018, but you could just see that you still have a lot of damage from Rhizoctonia inoculated plots compared to non-inoculated plots. And navy beans, like I said, you see a row of plants dying. Again, this is all caused by, early on the seedling post-emergence damping off because most of the root system is gone underground. Especially this can be also a problem if you have later emergence. If the soil conditions are not right after planting, then late emergence, some of these younger plants are more susceptible compared to the older plants. It's very a typical pattern for Rhizoctonia there. And corn, we have a lot of corn in rotation with sugar beets, especially in southern Minnesota there. But the tricky thing with corn, the Rhizoctonia, the AG2-2 can colonize and cause this brace root rot, but you don't see any impact on the yield for that. So what it's doing is the inoculum actually is getting multiplied year over the year, but you don't see any yield loss in corn. But when you go into the sugar beets, that's pretty obvious to see the yield loss because if you lose the root, you're going to lose the yield. I'll just show you some of the impacts here. Excuse me. The other challenge I showed you some fields with both Rhizoctonia Aphanomyces. This particular field, we were only looking for a field with a natural history of Rhizoctonia, and we planted. But sure enough, after a few weeks after planting, we realized not just Rhizoctonia, but a lot of Aphanomyces happening. This picture was taken on July 2015, July 21st. Right 2 weeks after I took the picture, there's hardly anything left in that. So that's pretty devastating to see there. Excuse me, I don't know what's happening with my mouse here. Yeah, that's right. And as I said, Sarcospora is another major problem that our growers are battling with. This is one of the growers' field. Typically, they're doing about 5 to 6 applications for managing this disease. In this particular grower, after this is during his third application, right about this point, he ran out of the fungicide and he thought, okay, well, I got 2 sprays anyway before that, and then he gave up on the spray. By the end of the season, the impact was pretty significant. So just with the lack of 1 spray, he lost about $373 per acre. So that's a tremendous loss for the grower. So in total, I think this one co-op, just because they got a lot of rain, and then we have a lot of fungicide sensitivity issues for Sarcospora reticulum isolates, and with all the heavy rainfall events, some of the contact fungicides were not as great, because the interval was very long, and then the efficacy was very short for them. They lost about $100 million, so that's a pretty significant problem. So I just want to jump into Rhizoctonia. So when we talk about Rhizoctonia, that's a Rhizoctonia solana we are dealing with, not ZA or anything else here. And the teleomorph is Thanetiforus cucumeris, right? Although it's not very common to see the sexual stage, but my predecessor has seen it a few times on sugar beets. And then there are multinucleates and binucleates for Rhizoctonia, and they're divided into several different anastomosis groups. And when it comes to sugar beets, we are mainly concerned about AG2-2 and also AG4, but AG4 is actually mainly for seedling damping off compared to the root rot later in the season. And within AG2-2, we have different intraspecific groups we call ISGs, 3B and 4. The big difference between 3B and 4, 3B could actually grow at 35 degrees Celsius compared to 2-4. And I'll just show you how it impacts, where we see that in different parts of the states. But when it comes to the root rot symptoms caused by 3B and 4, they're pretty similar. You can't really distinguish what's caused by 3B or 4. Again, these are some of the isolates that are actually collected from various samples. There are some 2-3Bs, some 4s. This is actually in AG5. It's not uncommon sometimes to isolate to some of the other AG groups too. So my predecessor did a big survey back in 2010, trying to see, okay, if we recover a lot of isolates from different parts of the state, what do we see? So the thing that they observed was in the northern end of the valley here. So this is the Red River Valley on the Minnesota side. This is the North Dakota side. So about 66% of the isolates were AG2-4. This is not able to grow at the higher temperature. Whereas most of the isolates, about 56% of the isolates recovered from the southern Minnesota were 2-3B. So this is able to grow at that higher temperature. But if you compare the crops in rotation in the south versus north, there is more corn in the southern Minnesota compared to northern. I think that's what is driving these 2-2 ISGs, the 3B ones, compared to the northern end of the valley here. And when it comes to Rhizoctonia, it causes pre or post-emergence damping off. More commonly post-emergence damping off, even though it's not uncommon to get the pre one too. Again, when the seedlings are very young and if the conditions are favorable, you can lose the whole seedling. As you go later into the season, especially the infection is underground, the best time to go out and scout for the fields is in the afternoon, because if you have enough moisture, you may not realize there is some infection underground. But I think really when it's hot and a little bit drier during the afternoon, you can see this wilting, some of the older leaves right away compared to the younger ones. Again, these are some of the inoculated plots that we have, pretty significant crown rot in this. Again, you don't see that in a grower's field. And by the end of the season, what we see is in a completely dead plant, we call these spiders, and all you see is some legs, right? These are the petioles that are lying flat on the ground to see the effect from Rhizoctonia. But really, I think that the most affected part is the roots. So you get these ladder-like lesions develop on these roots. Sometimes they're just on the surface, but sometimes they go deep depending on the infection level. But we're seeing something different now. Since 2009, we have Roundup Ready sugar beets. So the growers are not doing much cultivation to manage the weeds. They're not throwing any of the inoculum into the crown. So we are seeing more and more root rot developing just 2 to 4 inches or just below the crown, not really like a crown rot. So we put this question mark because we're not seeing much. Again, really good example. The crowns look really healthy, but again, the root rot is developing underneath. I think primarily the location of inoculum is actually driving this symptom development for these situations. These are the typical AG2-23B and then the 4 isolates that we recover from most of the sugar beet samples. And for some of the data that I will show you in my talk later, so we used a 0 to 7 scale. So this is actually an ordinal scale, not very linear, about 6 to 25% here, 25 to 50, 50 to 75%, more than 75, and 7 is a completely dead plant. So this is the sort of the reference that I'll be using for the future slides. This is how we rate the roots at the harvest. And the rhizoctonia is pretty bad because it affects the yield and also the recoverable sucrose. So what I have here is a 0 to 7 scale. The higher the root rot at the harvest, you're going to lose more yield. But I have 2 different varieties here. The blue triangles are the resistant variety, and then the red one is a susceptible variety. As you can see, the slope is pretty steep with the susceptible variety. The higher the disease towards the end of the season, the more yield that you lose. And the beets are harvested in October, right? They have to process them until June, which means they have to keep really healthy beets into the storage piles. Otherwise it's pretty nightmare for these factories to process these beets. The thing that affects most is the respiration rate, and also that has an inverse relationship with extractable sucrose and invert sugars. And here, the roots with a rating of 2 compared to 5 here. So the higher the root rot rating you have, the higher respiration rate, and then there's less extractable sucrose, whether these are 30 or 90 days after harvest. So it's pretty critical for them to do it. But one common practice, if they think they have a lot of rhizoctonia in a particular field at the time of harvest, they actually don't even bring them to the storage piles. It goes directly into the factory for processing. That's why they don't have to deal with the storage anymore. When we talk about, we can't just really complete a talk without this disease triangle, right? Really, we have this host and the pathogen and also the perfect environmental conditions. We have good resistance for rhizoctonia and sugar beet, but that comes with the yield penalty. And in terms of the genes governing resistance, I mean, these are only few genes, and they have really major effect on that. And for AG groups, I said 2-2 is predominantly, it's very severe for the root rots, even though 4 can be common too to cause some damping off. But in terms of disease development, I think it's largely driven by the amount of inoculum that's present in the soil, whether it's a very high or low populations. When it comes to environment, generally it loves the warm weather. The warm means the warmer weather for Minnesota, not for Louisiana or Texas. And the soil temperature, it has very significant effect. I'll show you later. About 4-inch soil temperature, we track that. I think it's critical for activity of rhizoctonia. Whenever it hits about 65, I think it's very active. When it comes to soil moisture, it does not need a ton of moisture, even 25% of the field capacity. I think that's pretty good for rhizoctonia to become severe in a field. Especially, if you have less moisture during the later part of the season, you may not see big development of the disease by the harvest. And some of the management tactics for rhizoctonia, most of the growers plant early, depending, if it's possible or not. And the crop rotation, although sometimes it's tricky, if you have soybeans and corn in rotation with sugar beet, even though you're doing rotation, you're not really helping for rhizoctonia. Some other options are resistant varieties, especially use of fungicides at the time of planting, whether it's a seed treatment or infra application or the post-emergence fungicides. So this is a particular study. We looked at the interaction of inoculum density and also cultivar susceptibility. So we have 4 different levels of inoculum: none, low, medium and higher rate. These are 60 kilograms per hectare. That's how much the barley infested Rhizoctonia solani was spread in the field. And then we used three different varieties: partially resistant one, and a moderate one, and a susceptible variety. So what we are looking at here is just at the plant stand in 2013. The conditions were really favorable right after the planting, so we could separate the different inoculum densities, especially during 13 weeks. And then whereas in 2015, not so great right at the beginning, but we got more rain from mid to late season, so there's a really big separation for the stands. And here I'm showing the data normalized to the uninoculated control plots there. And when it comes to these varieties, again, the resistant variety did pretty good. And then the moderate and susceptibility, they lost some stands there. Whereas in 2015, even though the conditions are not really favorable right at the beginning, we lost more stands. But again, the separation was really bigger by the time we hit the harvest. So this is just looking at the stands. So we have a paper in plant disease right now. It's in first look. So what we did, we used all the stand counts data from 3 to 13 weeks, and we actually created a new parameter. It's called area under stand establishment curve. So this really is showing the overall accumulation of the, let's say, the healthy stands that we have. So in 2013 and 2015, there's a pretty significant difference. It's much steeper in 2015. And there is no two-way interaction between the inoculum level and also the cultivar susceptibility here. So we have the partially resistant one and the moderate and susceptible varieties in 2013 and 2015. And depending on the seed company that we get, sometimes the moderate variety looks similar to the resistant variety, and sometimes it looks similar to the susceptible variety. But unfortunately, there is no way for us to know, what is it looking like. But just when you look at the data, that's what we're seeing there. In terms of effect of inoculum density and the cultivar susceptibility and the root rotating, there was actually two-way interaction in this particular trial. But again, in 2013, not really a ton of disease. Using the 0 to 7 point scale, we use a midpoint rating. And the resistant variety, really low disease, even with the 60 kilograms per hectare inoculum, very high inoculums, and a moderate and then susceptible one with a very high. We saw the very similar pattern here, but like I said, the moderate variety that we used in 2015 was similar to this resistant variety, but obviously the susceptible variety had really a ton of disease developing by the end of the season there. And the same thing we saw in terms of the yield, but in 2013 and 2015, where we had a higher disease development, we lost a lot of yield compared to the non-inoculated plots. The same thing was reflected in terms of the resistant or the susceptible variety for this. And the same thing for recoverable sucrose, the same pattern there. So in general, what we have seen that the inoculum density has really big impact on plant stands and fruit rot at harvest, and also the yield parameters, especially if the growers have a rotation like a soybean, wheat, and sugar beet rotation, because soybean is a good host, but at least wheat is a non-host. And then by the time they go into the sugar beets, the disease risk should be a little bit less. So here they're okay with even going with the susceptible or moderate varieties. Whereas if you have a soybean, corn, sugar beet rotation where everything is a good host for Rhizoctonia, they can't really go with a susceptible or moderate variety. They have to have a variety with a strong resistance there. And I want to jump into the seed treatments here. I showed you some of the sugar beet. This is actually the raw sugar beet seed. It's very woody, and it's a monogerm. And basically, it's pelleted. Every seed company has their own color, so you can use a vacuum planter to plant this. Something new since 2014 in the sugar beet world is actually to have these SDHI treatments. These are very effective for managing Rhizoctonia, as it took multiple years for field and greenhouse evaluations there. So finally, we have penthiopyrad, we have sedaxane and fluoxyperoxide. So these were labeled between 2014 and 2017. So since 2017, the sugar beet co-ops decided it doesn't matter, every single seed that's sold in Minnesota and North Dakota should come with one of these seed treatments, because the effect of Rhizoctonia was pretty severe early on. Even though we have some varieties that have genetic resistance to Rhizoctonia, it won't kick in until the beets reach 6 to 8 leaf stage. That's why it's very critical to have these seed treatments there. And in general, they work very well. I have my untreated control, the blue line here, and number of plants for 100 foot of row. And compared to some of the commonly labeled seed treatments, the emergence is pretty good here right out of the bat. And then as you go later, about 3 to 4 weeks, that's when the peak activity for the seed treatments, and they go down as you go towards the harvest. But again, the best production is right around the corner here, between 3 and 4 weeks. And if you're a grower, I think you would love seed treatment because you don't have to do anything, right? You buy a seed and it comes with the seed treatment. That's very useful for you. And then if you have 700 acres of sugar beet and you have 2 good days in April 15, right? What do you want to do? You don't want to get off the planter and then go cleaning your plug nozzles. So there is no dealing with the nozzles there. Like I said, the beets are really susceptible to Rhizoctonia early on, so it's better to have some kind of insurance for the farmers to do it. But the risk and the downside, sometimes growers think that, okay, well, if I protect it early enough, it's good, but it's really not. That's the risk. And I showed you some of the pictures earlier with the Roundup Ready sugar beets. Now we are not seeing any of the crown rot, but we are seeing more and more bottom root rot, right? But so how do we deal with this? I think one solution for this to look at the fungicides. So what happens here is, there is a tank and the fungicide that's distributed to this drip tube. This is your disc opener there. Now before the row closure, the furrow closure, you have this as a dribble for the fungicide. I know probably there is some practice for use of fungicides for soybeans and corn, but I think probably I would say sugar beet is probably a well-tested system for fungicides. And also the common practice for growers to do along with the fungicides is also a starter fertilizer. Typically they use 1034O just to help the growth and development early on, and availability of phosphorus. That's a problem when the soil temperatures are cooler when the plant sugar beets. But the big challenge is some fungicides mix very well with this fertilizer, but some will not, and they will plug the nozzles. So several different fungicides we have. Especially Quadris is very good for Rhizoctonia, but you can have some issues. You need to have good agitation. And Asteroid, it's a generic formulation of Quadris, but they made specifically so that it can blend very well with the 1034O. It's highly popular among our growers here. And we do have Xanthia and Elatus. This is a headline version with the biological, and this is actually a Quadris with another SDHA fungicide, that's Elatus. And looking at the efficacy of fungicides, they do work very well. Again, my untreated control from the trial there, and the fungicides, really about 200 plants compared to untreated control. And then once you get the good, decent stand, when you have applications, they tend to maintain those stands. You don't lose just like a seed treatment, there where it was going down as you go later into the season for that. The benefit with the seed treatment, we are just protecting the seedling, whereas here you're sanitizing the furrow, right? Putting in the seedling and also the soil. The protection is a little bit longer for 8 to 10 weeks, but the risk, it comes with a little bit risk, because when I said the soil is cooler and the drier conditions, you can expect to see some phytotoxicity. And if you don't have a good agitation system, you can have plague nodules, which is pretty tough. And the seed and fungicides, this is from 2015. When we have a excellent disease pressure, we can expect to have more disease by the end of the season with this, just with the seed treatments compared to in-furrow. The same thing we see in the yield and RSA. Again, definitely the in-furrow treatments are better under severe conditions compared to the seed treatments. Whereas some years we don't have really good disease pressure, everything is inoculated, in the same way, just because we don't have the moisture. By the way, our average rainfall during the season is about 12 inches. And there are some years we get 8 or 9, so that's pretty dry. We don't get enough disease development there. So you don't see any difference between them. As I showed before, that 4-inch soil temperature used to be a key factor to do a post-emergence application in sugar beets. But now since 2017, when we have these SDHI seed treatments, things have changed because we do have some protection early on. Prior to that, if you do a later emergence, the post-emergence application, it was not really beneficial for managing rhizoctonia there. So some of the data here. So what we have been doing is testing 2 different types of application, especially as a post-emergence band application here. So we do a 7-inch band. It's a 4 and 8 leaf application and trying to see which will work better. But the overall results are, both are better actually, 4 and 8. So it doesn't matter when you get. So now there's a larger window for the growers to do these applications. So when we looked at this trial again with the 2 different varieties, there's a huge impact on the susceptible variety, right? And of course, it's very conducive for disease development. Here, there's no post-emergence application versus 4 and 8 leaf application. There's hardly anything in the resistant cultivar with this post-emergence applications, whereas susceptible, there's a big difference. I mean, again, the 8 was much better because probably a little bit later, it's actually giving some of the benefit going into the season. Again, same thing for the root rot incidence. And we did see some interaction with the variety and the post-emergence application too, right? So the resistant variety, it comes with some yield penalty. When we had this post-emergence application, there was a little bit of yield bump for this. But the susceptible variety, where without any post-emergence application, about 26. And then once we get this 4 or 8 leaf application, there is a huge benefit in terms of the yield boost for this. So whenever the growers are using the susceptible varieties, I think it's nice to have this post-emergence application. And also the interaction between the at planting, whether it's a seed treatment or in furrow, and the post-emergence application there is nothing on the seed. You put 4 or 8 leaf application, there's a big boost in yield for this. And just with the seed treatment, and when you add a 4 or 8 leaf application, you can see a big benefit. Compared to if you have in-furrow application and a seed treatment, even if you do a post-emergence application, you don't expect to see a big benefit because the protection is lasting for 8 to 10 weeks with this to do that. So I think my watch is going too fast here, so I got a lot of content to cover, so I'm not going to go through the summary. But in general, I think the seed treatments are pretty effective early on to protect these stands. But post-emergence applications, especially if the conditions are favorable later in the season, just like if we get a lot of rain in July and August, that's when we see the most benefit. Otherwise, we are not seeing much of the benefits there. Since we have these brand new seed treatments with that, the next thing to do was probably to get some baseline sensitivity because, going two to 3 years ahead, and if things are not working, we could look back and say, okay, what has changed for this sensitivity for this? So that was part of one of the objectives for Petiba. And also we used the Qol fungicides and we looked at if the EC50 has shifted between baseline and non-baseline isolates here, because we have been using azoxystrobin since 1999, and then pyraclostrobin, that's headline, since 2002. And we also use, since soybeans or sugar beets are grown in rotation, so we also got some isolates from soybean. And then we looked at, okay, whether it's EC50 is any different between soybean versus sugar beet isolates. So we used 35 isolates in total from both soybean and sugar beet for this particular study. Again, this is pretty standard procedure for a poison plate assay. Just amend this PDA with different technical grade formulations, whether it's SDHI or Qol fungicides. We do know that there is alternative oxidation for Rhizoctonia Aphanomyces, so we amended with sham for this. And then these were cultured, these are transferred to the PDA, and then in dark for 48 hours, and the 2 axes were measured. And then we got the EC50, the effective concentration, to reduce the 50% growth compared to non-amended control. So, for example, one of the isolate, there is highest concentration, 10 micrograms per ml, and then non-amended plates. So we can see a very good difference in terms of the inhibition here. The overall observations from SDHI fungicides, whether it's sedaxane or panthiopyrite or fluoxychloroxide, the EC50s, these are again in micrograms per ml, pretty low actually, whether it's a soybean or sugar beet. It's 0.11 and 0.099 0.19, 0.14 for fluoxyperoxide. But thanks to statistics, right? The things are very small, but very consistent. So there is highly significant. But in terms of biological significance, there is no concern at all for this SDHI fungicides right now. And in terms of the correlation, when we looked at the three fungicides with the same mode of action here, there's pretty good positive correlation, although ranges from 0.32 to 0.34 when it comes to R, but it's a highly significant correlation. It's all positive. So we do know that SDHIs can have some cross resistance, but eventually I think if we see a breakdown in one, then probably we have to check for the other two fungicides too for this. And for QI fungicides, most of the work that was done with pyraclostrobin was straightforward. Azoxystrobin was a little bit tricky. I'll just show you probably in the next slide there. Again, between soybean and sugar beet isolates, there was no statistical difference between soybean and sugar beet, whether it's for pyraclostrobin or azoxystrobin. Again, these cultures were again all the way up to 2017. And when we looked at the QI fungicides, because we do have baseline and non-baseline, especially for pyraclostrobin, that's prior to 2002 was considered as a baseline, and everything else was a non-baseline. And then there was no difference between baseline and non-baseline isolates for pyraclostrobin. The tricky thing with azoxystrobin was it's very hard to get the perfect EC50. It seems that even though the growth on the PDA plate amended with azoxystrobin, especially with the technical grade, there was inhibition, but in terms of the growth on the plate, it's not very clear. There was really sparse growth if you increase the concentration. So it's a big challenge to determine the EC50 directly with the technical grade. So out of 35, only 22 isolates got the EC50 values, but based on the 22, again, there's a significant difference between experiment 1 and experiment 2 for the mean EC50 values. That's why these data was shown separately here. But between prior to 99 and after 99, there was no difference whether it's 1 or 2, and it's 0.07 for azoxystrobin. So right now, basically, we don't have any concern in terms of fungicide resistance development for QI fungicides. So for summary, I think the SDHIs have extremely low EC50 values, which is very good. And also, that's how we established the baseline sensitivity for these. For pyraclostrobin, they're about 1 microgram per ml, not as low as SDHI, but not as high either. For azoxystrobin, it's about less than 10 micrograms per ml. But again, we have not seen any failure at the field level for azoxystrobin. Like I said, there are a lot of issues with azoxystrobins to do the EC50 inhibition studies. I think we need to devise probably a mycelial, maybe a broth-based assay or some kind of in vivo fungicide sensitivity assays just to determine accurate EC50 for azoxystrobin. Talking to some of my colleagues at Nebraska and Michigan, even from Syngenta, everybody has the same problem with azoxystrobin. And I want to just quickly jump into this detection of Rhizoctonia solani with qPCR. So like I said, most of the sugar beets are grown in a 2 to 3 year rotation. But if you have soybeans and corn, even some of the weeds that are common in sugar beets, these are also good hosts for Rhizoctonia. So there's always this inoculum bridge in any given year. So by the time you go into the sugar beets in year 3 or 4, you're going to see a tremendous impact on yield and also losing quality. So the traditional way is to bait a particular soil and then plant some seed and wait for 4 weeks and then see how many seedlings are dead or alive and then rate the remaining seedlings to get a 0 to 100 index. That's called root rot index. It takes 4 weeks and it's very time consuming. So we thought, okay, how about just probably develop an assay based on DNA detection, which is very specific, and also will let us see if we can quantify, how much Rhizoctonia is present in the soil. The ultimate aim for this soil was to kind of get the risk assessment, right? When you say that, okay, you get this much CT value or DNA, you could say that that's a low risk soil. So they could probably go for a shorter rotation, or they can have probably the crops of their choice compared to high risk soil, where they may need to have a lot of small grains or extended crop rotation for that. So this is how the typical growth chamber assay looks like. 25 seed in 4x4-inch pots, about 4 weeks. But one thing that we do, we remove these plants, the dying seedlings, every alternate day, because if you keep them in the pot, artificially you're actually increasing the inoculum in the pot, so that may not actually represent what's happening there. And we did some of the pilot experiments with infesting soil artificially with the sclerotia of AG2-2. But the challenge here, even with the 1 milligram per kilogram soil, we got about 67 in the root rot index. Once we go to 10, we lost every seedling by 12 days. So that's a pretty significant. You can't really separate the soil 1 versus 10 milligrams per kilogram of soil. And anything about 10 was 100%. I mean, most of the plants were dead within 2 weeks. We did not develop any primers or probe for this study, but we also used from the existing literature from Burge et al. It's based on ITS. And one thing that's unique to Rhizoctonia is also it's a multinucleate. Each cell has about 8 to 10 nuclei. So ITS itself is a multi-copy. And then if with this multinuclei, the detection is pretty specific, and you can actually detect very low amounts. The only downside for this assay was it has some cross-reactivity, the specificity to AG2-1 TABAC isolates. I don't even know what's this TABAC, but we know for sure that AG2-1 is not a pathogen for sugar beet, so we're not really concerned about that. And we tested 2-3B and 2-4 with a serial deletion for 10-fold serial deletion with the DNA there. The limit was about 1 picogram, because once we hit below 0.1, and that was out of range of detection for the Roche light cycler. So that's about 1 picogram we could detect with this assay. And the CQ value, and the other big challenge working with soil-borne pathogens, right? I mean, so what kind of soil sample that you use for DNA extraction, whether you use 100 milligrams or 5, right? So how do we represent that variation? So we compared 2 different kits, and then we decided to go with the 5 grams. That's a little Power Max. It's expensive. It costs about $25 just for the kit itself. But we got a good separation, and also, looking at the 10-fold dilution series for DNA, it's not close to perfect 3.33, but it's a pretty good representation for the soil dilution. So most of the samples were processed with the 5 grams of soil. So we did sample individual fields for this particular project, about 16 fields in our growing area. In each field, we actually sampled at 5 different spots. And then we also selected outright, visibly diseased spots and also healthy spots. In each spot, we actually sampled 1 soil course from top 2, 2 to 4, and 4 to 6. We separated this soil course because we just wanted to see, what's the depth of this inoculum that we're seeing for this. And from where we got the samples, we also got soil for the growth chamber assay. That's about 1 gallon. And we got the root rot index score. And from the same spot, between those 2 rows where we sampled the soil, we also rated 10 roots on a 0 to 7 scale, right? These are all different parameters that we have. Then we could actually try to put these pieces together and see what we are seeing. So looking at the soil index on x-axis here, 0 to 100 scale, this is based on the growth chamber. And this is the actual root rot rating that we saw in the field on 0 to 7 scale. Well, not really, good positive correlation, about 0.34 R square. But our R is 0.59. But again, we can see when we see in the growth chamber, I think there's either very low or very high. We did not see many samples with the medium root rot index. And this is here, I have the root rot incidence here. Anything that's greater than 2 that was scored here. And then the soil index based on the previous slide, that's on 0 to 100. Again, there's a positive correlation that's highly significant there. for the root rot index versus root rot incidence, right? Now it gets a little tricky. Now I'm just showing the data from the soil DNA. These are based on a 1-inch cores. There is no DNA that's visibly detected with the qPCR, but we have a ton of root rot index developing in the growth chamber. I mean, we do have some spots, but most of these are located right here. So this speaks to the distribution of Rhizoctonia solanae in sample. I think it's pretty tough to get 1 or 2 cores and then try to represent that the amount of Rhizoctonia in the soil there. And here again, the correlation of the soil DNA, again, this is all 6 inches here. And also the final root rot rating, that's pretty similar to the other one. But in addition to the distribution of Rhizoctonia in the soil, I think there is something else going on too there. Because we know predominantly, I think AG2-2 and AG4 can cause damping off. But a study from 2010 by Melvin Bolton's group, there are some other AG groups, especially in the greenhouse, they could cause seedling damping off, but we don't really know whether under the field conditions, if the diversity of the AG groups exist in Minnesota and North Dakota with the sugar beet production, or if they're able to cause damping off just like that. Because in a growth chamber, we are maintaining this perfect 25 degrees Celsius during the 4 weeks period, which is not possible to have under the field conditions. So that could be another reason that's overestimating the value there. And they also looked at the mature root rot with several different AG groups, but predominantly 2-3B and 4 are causing the root rots. But you could see some could cause probably 1 to 2. So when we sample these full fields, probably there's a possibility that some of the lower root rot is caused by other AG groups in those fields. So in general, I think some of the observations from this study, there are more samples that are positive in the top 2 inches compared to 2 to 4 and 4 to 6. Again, this also speaks to the type of cultivation and land preparation that's the growers do, because nobody does any moldboard floor. Most of the top 4 inches soil is being worked out. That's where we are seeing the inoculum. But the biggest observation from this, when the fields with the severe Rhizoctonia, the inoculum was distributed throughout the top 6 inches, where some fields with the low and moderate, we are only seeing in the top 2 or top 4. There is no clear pattern for that. Again, like I said, some of the root rot index coming from the growth chamber, it could be caused by other AG groups, which this assay is only very specific to 2-2. And I think probably the soil sampling needs to be more something like a grid sampling, or there should be more cores so that we can actually represent what is happening in terms of disease distribution and then possible risk for the field. So how am I doing with time, Leonard? I think, am I over my time here already?
Yeah, just by a few minutes, Ashok. So
okay.
We'd love to ask some questions if you have time. Yeah.
Yes, so I apologize for this one too. I got a lot of content here. I'll just quickly only show maybe one for this particular project. The other thing that's very important for growers is to detect Rhizoctonia. Like I said, unless you go in the afternoon and you have perfect soil conditions, there is no way for you to know there's root rot underground, right? So it's very good to know if there is any way we know Rhizoctonia root rot. And number 2, it's very useful for the sugar beet co-ops to know the fields that are affected with Rhizoctonia, because that's very helpful with the harvest operations. They don't have to take these beets to the factory. They can open these before even the main harvest, and they can process these into the sugar. So that's the main idea for this particular project. And the other thing, if you look at the 0 to 7 scale, we don't see any visual symptoms on the tops, just at least until 3. Once you hit 4 and 5, it's very easy to see the wilting on the tops, but 4 can be tricky too. If you have good soil moisture, you don't see any of that. So the idea for this project is, can we detect this Rhizoctonia for these 3s or even 2s, early in the season, so we can actually intercept with some kind of fungicide management for this. So if you look at how the plants reflect light, if you have a healthy leaf, they absorb most of the red and blue, reflect some green, but a lot of NIR is being reflected. When it's stressed, there's not much NIR coming off of those. And when it's a dead leaf, everything looks mostly flat for that, right? So that's a basis for the reflectance studies. So what we did, we actually inoculated these plants at several different levels of inoculum. And also sometimes we place individual barley grains next to these roots. And we did a time series sampling. And what we used is a handheld spectroradiometer. Basically, it's called a leaf clip. You can actually detect the wavelengths all the way from 300 to 1000 nanometers with this sensor. So that's what we did. Over 7 time points, we got about 3,500 individual plants. This is how the wavelength pattern from one infected leaf looks like. There's a lot of noise on the lower end and higher end. So once you trim it, it looks much smoother. Looking at all the data points, about 1.8 million, we ended up with 2 wavelengths that's highly correlated with the Rhizoctonia root rot, just about 689 and 698. So our hope now is actually to get some sensors very specific to these 2 wavelengths. And then if we fly a field and see if we can detect Rhizoctonia or not, because a lot of times there's a varietal difference. There are confounding effects from other stressors like abiotic stress or insect stressors. So that's why we want to separate this. And this experiment was done in this particular way. And we also want to combine thermal imaging into this visible and NIR spectrometer there. With that, I would like to thank my team members and a lot of collaborators for other projects and a lot of undergraduate students and the funding sources for this. Again, my apologies for going over time, but I'll be happy to take any questions now.
All right, thanks, Ashok. So if you are able to stick around and ask some questions, that would be great. I would just maybe ask if you're willing to turn on your videos so that Ashok actually realizes there's real people behind these little squares with our names. So yeah, so if you can stick around for questions, yeah, that would be great.
Ashok, I've got a question for you.
Yes.
The whole subject of in-furrow and seed treatment and post-emerge treatment, those all relate to Rhizoctonia, right, on sugar beet?
Yes.
Okay. Have you or anybody else done economic studies? I realize that everything you said, that the outcome could be different depending on the disease pressure and variety susceptibility and all these other things. But has anybody ever tried to gather some of those factors together and do an economic analysis to see over the long term what would be the better bet? It's obvious that seed treatment is more convenient, but is it more cost effective?
That's actually, I think if you're a grower, I think that's the main thing, right? You want to answer, right? What does it cost me? How much I'm making? But relatively, since seed treatments are new, it's not being done, but we know how much they cost. But one thing the growers are thinking, it's been 5 years and they're happy with the seed treatments the way they are. And now they're seeing more of the bottom rot, like I showed, right? We're not seeing any crown rot. They want to do in-furrows. But once they saw the in-furrows, which are actually having 8 to 10 week efficacy, now they want to go back to the table and say, hey, we don't want to pay anything for the seed treatments. And that's what they are thinking. I don't know why I want to pay for both to do it. I would say, in generally, it's very beneficial to have either a seed treatment for in-furrow, especially if you have a susceptible variety, because you can protect a lot of yield. Because when the growers are buying the seed, the cost is same, whether it's a resistant or susceptible one. But they're always looking for the susceptible one because they're hoping to get that, like, they can protect it somehow and do it. So.
Okay, thank you.
Thank you.
Other questions for Ashok?
Ashok, I have a question for you. Most of the fungicides that you use, are those mostly systemic fungicides that circulate throughout the sugar beets?
That's a very good question. So when we talk about it, we are trying to deal with the root disease, but we're actually applying the fungicide on the tops, right? So, you know, QOIs are locally systemic. I mean, they're not going to move throughout the plant. But the key is, if you look at the label for Quadris, what they say is you just need to get those crowns covered. Typically, even though you're doing the post-emergence application, I think the key is to get some into the soil that's surrounding the roots. And if you compare all the QOIs, you know, Quadris has the best activity in the soil. I don't know why. Probably maybe somebody from Syngenta knows the answer, but once it gets in the soil, it has really good efficacy compared to even pyraclostrobin too. But back to the question, no, these are just locally systemic, but I would say the activity is really good in soil, especially for Quadris. I think that's why it works well with Rhizoctonia there.
So you don't have much risk of it residing and staying within the sugar beet itself after harvest and through all the processing?
No, don't think so. It persists well in soil, but not in the sugar beet, I think, especially, you know, the residues are not that harmful. So.
Ashok, I actually have a question too. So at the beginning of your talk, you said that, you know, there's Rhizoctonia and there's the Aphanomyces, which was the kind of the second most important pathogen. Is there any known interaction, like a synergistic or some other type of interaction between the two, or when they co-infect, you just add the, you know, the damage from both?
Ideally, I think you can get damage from both. We tried to do some experiments, but either one kills and the other has no chance, or sometimes one starts the infection and the other gets in, and it's very difficult to tease. I mean, what's doing what when we have the mixed infections, Leonard.
Okay.
But one thing we can say for sure, I think the moisture drives, especially in a grower's field. If it's really dry season, and if they have both, I think Rhizoctonia will be the one on the top that's rising to the occasion and then doing the damage.
Okay. And so I guess as a follow-up, you know, like Mark said, the treatments that you mentioned, the seed treatments at least, are target the Rhizoctonia. So is there anything that's done to manage Aphanomyces?
Yes, actually, we have a very good seed treatment for Aphanomyces. That's tachagarin. That's a hymexazole. That works like a charm. But again, the efficacy only lasts for 4 to 5 weeks after planting. But the other thing, it's a kind of serendipity that we found in the sugar beet world. One of the byproducts from the sugar factory, it's called it's precipitated calcium carbonate. We call it waste lime. That actually works very good for managing it. It's good for 10 to 12 years once you put 10 tons of waste lime in a field for Aphanomyces. You know, when you apply lime, it increases soil pH, but naturally our soils are already at 8 pH. We don't need to increase our pH. You know, if you go to France, they have a lot of Aphanomyces in peas and then sugar beets. They have low pH soils. Our soils are already high pH, but still when we apply lime, that tremendously helps with Aphanomyces. That's one practice that our growers do, actually.
Okay, thanks. Any other questions for our speaker today?
I'll be happy to take any questions by email if you have anything. So.
All right. Well, thanks for the offer, and thanks for speaking to us today. So we'll let you go. We wish we could give you a round of applause that you could hear, but we're kind of.
But thank you very much.
Ashok. Appreciate
We
Good talk.
appreciate your time. Yeah.
Thank you.
All right.
All right.
Bye, everyone.
Bye.
Bye. Well, thanks so much again.
Thank you.
A lot of great information.
Sorry, it just the time was going over. So
I understand it's hard to pick out the
do that.
When you have so many exciting projects. So are you in tender track? Or are you going to go?
I'm just going to submit my package now.